Before we talk about cells, let's talk about how things tend to move in our universe. Our universe is expanding, meaning that it is stretching apart. And so molecules in our universe tend to try to get away from each other. Take, for example, these little green dots. We could pretend that these represent fragrance molecules from cologne. If you were to spray your cologne in one corner of your room, you might be able to smell it at the other corner of your room. This is because these molecules will bump into each other, like little billiard balls in a game of pool. This means that they will space apart, seeking to get further and further away from each other. Molecules do not like to be in a high concentration, and so they seek an area of low concentration. In other words, molecules do not like to be crowded. The direction that they move in order to stop being crowded is called the concentration gradient. A concentration gradient is a flow of molecules. That flow typically has both a direction and a strength. If this is a membrane, and these X's are molecules, then the concentration gradient would be to the right. This is because the left is more crowded than the right. Gradients always move from an area of high concentration to an area of low concentration. This gradient has both a strength and a direction, a lot like a river. This flow can be very strong if the difference in concentration on either side of the gradient is large. It could also be slow if that gradient is weak. And so to make this gradient weaker, we could allow flow. We could say that these two X molecules are now on this side. This would reduce the strength of the gradient. Whereas if we moved all of the molecules to this side, that would make it stronger. The flow is stronger when the difference on either side of the gradient is bigger. The direction is determined by which side is higher and which is lower. What determines the direction of that river, of that concentration gradient? Concentration gradients flow from an area of high concentration to an area of low concentration. Concentration gradients are like a river in that they have a direction and a strength. You could see that the direction here of this river is going this way. This river could be very strong, or it could be very weak. It depends on how much water is flowing. A concentration gradient can have a different amount of strength as well. This concentration gradient is relatively weak. There isn't much difference between the left and the right side. However, I could increase the strength of this gradient if I add more to one side. By adding more of these X molecules, I have increased the potential energy. If allowed to flow, this gradient will be much more powerful. We will learn later when we talk about mitochondria and how it makes ATP that storing energy in a gradient is very important for cellular activity. To increase strength, we must flow against the gradient. By flowing against the gradient, we increase its desire to move, and therefore its strength. Now we will talk about cells. Cells are full of stuff in their cytoplasm. There's also a lot of stuff needed in our bodies outside of our cells in what is called extracellular fluid. This stuff, whether it be water, salt, sugar, electrolytes, or gas, creates a gradient. That gradient has to flow for a cell to survive. For example, oxygen. Oxygen must move into the cell in order for the cell to survive. Here we can see a concentration gradient. When you breathe, oxygen is carried by your blood cells to your other cells in your body. This cell could be a fat cell. In order for it to survive, oxygen must flow in. And so the outside of the cell will have a high level of oxygen, and the inside will have a low level of oxygen. Because of this, oxygen will naturally flow into the cell along its concentration gradient. To reduce the strength of a concentration gradient, you move with it. Much like this man is flowing with the river, when you are a molecule that flows with the concentration gradient, you are engaging in what is called passive transport. This is also called simple diffusion. Simple diffusion is a type of passive transport in which molecules move straight through a lipid bilayer with no energy needed. Just like this man in the lazy river is not spending any energy, nor will an oxygen molecule in order to flow through a cell membrane. Cells will continue to move molecules along this path until they reach equilibrium. And so here we can see molecules of orange dye, and they will move through the cell until both sides are equal. It starts with one side being high concentration and the other being low, but then it ends with both sides being equal to each other. At this point, there is no longer a single direction that the gradient flows. Rather, it flows in both directions at equilibrium. Your lipid bilayer is made of small fat molecules called lipids. Your lipid bilayer is called a lipid bilayer because it has 1, 2 layers. These small lipids do not let everything through. Sometimes molecules can move straight through. This is simple diffusion, like in the last slide. But sometimes molecules are too big, and they need assistance. They need a tunnel. A tunnel does not push them through. There is still no energy required. A tunnel, like this tunnel here, is simply an opening with the correct shape for a certain molecule to pass through. Much like this kayak here can go with the flow of this river to go through this tunnel. The person in the kayak does not even need to paddle because he is going with the flow. He does, however, need to go through the tunnel. Simply smacking into the bridge will not work. This is something that we see in our cell membranes when we look at water. When water tries to move through our cells, that is called osmosis. Water is an example of facilitated diffusion. It requires a special protein called an aquaporin. Sometimes a concentration gradient is moving the opposite way that we want to move things in and out of our cells. And so we must spend energy in order to move against it, like this swimmer is swimming upstream. We call this energy ATP. We must spend ATP in order to move materials against the gradient. And so perhaps in this picture we will use our X molecules again. And we can say that this is the cell membrane. And here we have our lone swimmer. We must use a special kind of protein to pump this molecule against its gradient. So I'll try to draw that. I'll use a different color. We can use pink. This protein will grab this X molecule, spend ATP energy, and throw it against the gradient. This will increase the strength of the gradient and therefore potential energy. There are many types of active transport. The first one I'll cover is called phagocytosis. Phagocytosis is when cells use what's called pseudopods in order to eat. Pseudopods are extensions of cytoplasm that allow organisms to either move or eat. I'll show you an example. Here we have an amoeba. Amoebas are single-celled organisms that eat other single-celled organisms like the ones here. Amoebas are like the lions of the microscopic world. They eat everything, and I'm going to show you how it eats these unfortunate paramecium.
Con los terroristas.
Nice little throwback to, 2010? Anyway, so yeah, that was that was an amoeba eating 2 paramecium to, what is that song from? What is that song? Harlem Shake? Anyway, I thought it was cool. So this thing is the amoeba, and it is eating these paramecium, and you could see that, let's play it again. You can see it right here. These are pseudopods. These pseudopods stretch around until they trap their prey.
Los terroristas.
Then right here, these 2 pseudopods fuse. And now that it has trapped its prey, it will start to squeeze. And they know they're trapped, so they try to escape, but it is too late. The amoeba has them. I want to mention another type of active transport called pinocytosis. We just talked about phagocytosis. In phagocytosis, there is pseudopods. So, pseudopods. In pinocytosis, there are no pseudopods. Instead, pockets are created with pressure from outside environments. And so liquids get trapped in these little bubbles. They fuse right here, and they create a small vesicle. This vesicle will then travel throughout the cell to its final location. On the next slide, we will talk about receptor-mediated endocytosis, in which tiny little receptors are used to do the same thing as pinocytosis. Pseudopods are not always used during active transport. Sometimes we use receptors instead. And so in receptor-mediated active transport, there are no pseudopods used. Instead, vesicles, like we just saw. Vesicles are all that is used, with receptors. Receptors are usually carbohydrates, although they can be a mixture of carbohydrates and lipids. These carbohydrates sit on the lipid bilayer, where they are ready and waiting for molecules to fit into them like a lock and key. This creates an address for the cell. And so receptor-mediated transport is a lot like having an address where parcels are delivered. Endocytosis is when we take those parcels into the cell, and exocytosis is when we send them out. So endocytosis is in, exocytosis is out. So now we have talked mostly about diffusion and how molecules like to move to from an area of high concentration to low. They will disperse. Water follows these same rules. However, it's not always so obvious when you look at it. Instead, it is easier to think of water as moving toward solutes. Solutes like salt or sugar, or ions found in electrolytes, like in Gatorade. The reason why we drink Gatorade is because when we pump salt and other electrolytes into our cell, water tends to follow. And so here we can see these green Gatorade molecules have gotten into the cell because we drank Gatorade. And so water will follow it. Water is a follower, and that is because it seeks an area where it can be less crowded. In order to be less crowded, it moves towards other solutes. Water is a follower. Water is like any other molecule. It will move from an area of high concentration to an area of low concentration if it is allowed to through a membrane. For example, here we have a membrane, and here, and this is a time lapse. Water moved from this side to this side because, well, it went through, because on this side, water has a high concentration, and on this side it has a lower. It might look like it is even, but it is not. Because on the right side there is these little sugar molecules that is displacing the water. Much how ice displaces water in a cup. If we had two cups, If we had two cups, and this one was full of water, and this one had ice in it, and water, which cup has more water? Well, obviously this one does, because this one has ice displacing the water. In a similar way, this side has more water, and this side has less. So water will move where it can be less crowded. This makes it look like water follows solutes. So it might be easier for you to remember that water will typically just move to where there are more solutes. And so that's what people like to remember. Water follows solutes in osmosis. The truth is, it is following the same rules as everything else. You are mostly made of water. And so cells live in extracellular fluid. This is the fluid outside of the cell. That fluid has to be just right, or the cell can't live. What do I mean by just right? The conditions have to do with solutes, like, for example, the appropriate amount of electrolytes. A cell that is in the wrong solution is not in homeostasis. In order to be in homeostasis, a cell must be in an isotonic solution, where the solution has equal levels, or rather equal density, of solutes as the inside of the cell. This is the perfect place where cells can have perfect osmosis. And so water will flow into and out of a cell equally in an isotonic solution. Equally. The flow does not stop. It is always moving, but equally, in and out. Hypertonic solutions is when water rushes out. This is generally because there are lots of solutes outside of the cell and very few inside. Because there are very few inside, water will move out, because water follows solutes. The other situation is hypotonic, where there are a lot of solutes inside the cell and very few outside. This would make water rush into the cell. This is Isotonic solutions are very important for maintaining your health. So, for example, if you exercise, you should have electrolytes. The best electrolyte that I know of is pink Himalayan sea salt. Better than Gatorade. I put a few little pinches of that in my water before I swim, as an example. So, isotonic solutions are like this IV. This IV drip is not pure water. It has water, but it also has salt and other electrolytes. Electrolyte means ions. Salt is a natural ion. When it breaks apart, it creates ions. Cells need to be isotonic with the solution that they are in order to be healthy. And so this IV drip to its patient must be isotonic with that patient's cells.